Okay, thanks for this give me this opportunity to present our works on this applying this structural system pharmacology to precision drug rescue and drug repurposing. So this drug action is quite a complex process. We still know little about how drug works and why this we have to see this kind of several talks about why these different individuals have different drug response. So currently the drug discovery mainly follow this kind of one drug, one genes approach. Actually this have little success, but you consider this when a person take a drug, actually, because these all proteins, if you consider the structure and their evolutionary relationships, a lot of them actually is related. So it's a drug is rarely interact with only one target. It's could they have a lot of off target. Actually, we don't know which one is interact. Also, this target is not acting independently. They interact with each other and also other molecular components in this whole biological network. And then this kind of information, drug-target interaction, will transmit to other genes. Actually, you can observe it from multiple omics, this kind of data set. This kind of this kind of molecular phenotype will directly related to this therapeutic effect or side effect. So these mutations could be changed these drug-target interactions directly, or this kind of drug-target interactions, or rewire this molecular network, or it could be regulated this gene expressions or other this functional states. So we believe in order to understand how drug works, we need a multi-scale modeling approach. We first need to understand how drug target interaction is understand this binding-unbinding kinetics. Actually, this so far this have not paid enough attention to this kind of problem, because for example, in this kind of anti-cancer drug discovery, there are a lot of talks about this cell-based assay. So this IC50, this measured in this kind of equilibrium conditions. But in human body, this drug-target interaction is never can reach this equilibrium condition. It's always non-equilibrium. So this major driving factor about this drug side effect or efficacy is K-off, this unbinding event. Okay, so in order to correlate this kind of in vitro assay with in vivo drug action, we need fully understand this kind of binding-unbinding kinetics. But if even you know this kind of drug-target interactions for single target is not enough, as we mentioned, we need to identify all possible this kind of drug-target interactions, and then map this all drug-target interaction into molecular network, see how it affects the whole system. Finally, maybe you can have some ideas how this genetic or epigenetic modifications or change this kind of dynamics of target binding or system response to relate it to this final drug response. For this purpose, we developed a series of techniques to model this drug target binding kinetics, identify this genome scale of targets, and also study this, characterize this dynamic rewiring of a biological network upon drug binding. So based on these techniques, we can repurpose drugs. Also, we can rescue these failed drugs. Okay, so here I'll give example. So the CTP inhibitor actually now is quite hot in pharmaceutical company. They think this kind of inhibitors will increase this HDL and lower HDL, because people think this higher HDL is correlated with the healthy heart, right? So several pharmaceutical companies spend a lot of money try to identify this, found these CETP inhibitors as new drugs, as a new blockbuster for after this Lipton. So Pfizer actually spent about $1.5 billion to develop this kind of CETP inhibitor called torcetrapib. Unfortunately, this drug is not increase this healthy heart. This actually it causes a heart attack due to this hypertension. Eventually, it's withdrawn from phase 3 clinical trial. So we want to understand what happened for this drug, why it caused this hypertension. So we start is this primary target. That's actually CETPs. This how design these drugs, target this target, right? Then we identify all possible off targets for this drug on a genome scale. And then we also construct this genome scale metabolic model, actually it's a kidney model, by integrate multiple omics data, then map this off-target binding with also this genetic mutations, see how this drug-target interaction and also mutations collectively affect this blood pressure. So then we can see what happened, why this drug, why it works on certain person but not cause deadly effect on other persons. So here are several interest findings. So we associate with this kind of drug target of interactions with some this kind of genes. This gene is not drug target, okay, just this payloads in this network. So if persons with this kind of deficient genes, it's okay if you don't take this drug. If you have this normal genes, this drug also safe. Only you have this kind of both of them exist. They have this synthetic lethality. This cause this kind of side effect. So in the remaining talk, we'll give a brief introduction on this kind of algorithms to predict this genome-wide high-resolution molecular interaction models, including both this protein-ligand interaction and protein-protein interactions. So this representation is kind of problem. The formulated problem actually is this kind of bipartite graph. One side is a representation of structural proteome with similarity between these proteins. Another is chemical genomics data, have this kind of similarity between chemicals, right? If you know how associations between them, you link them, and then you predict new interactions. So this kind of formulation actually is quite standard. It's not new. But the difficulty is how to measure this kind of similarities between proteins and also between chemicals. So now if you look at literature, this similarities basically is based on global structure or sequence similarity or gene ontology, protein-protein interaction. But this kind of measurement don't really capture this context-specific information about these interactions related to drug target interaction, because I've showed before, these mutations, this concentration, post-modification, or this oligomer states, all will change these bindings, right? So we need this kind of context-specific similarity between them. So we developed this kind of new algorithm, can measure this similarity. It's in a context-specific, also this can crowd for space. Basically, if two proteins have totally different sequence and structure, we can still measure their similarity between them. The basic idea is to determine this protein binding interface and how to measure their similarities. Then we also have this kind of new graph mining algorithm. We call it the enrichment of network topology similarity. The basic idea is to use this, we implement this scalable single source k-diverse shortest path algorithm to explore this network. Actually, it's much better than this random walk with restart. It's, for example, PageRank is have applied a lot in this kind of problems. Also, statistical framework to measure this statistical significance of ranking. Not just like page rank, just give a rank. We also give you p-values and q-values. Okay, besides benchmark, actually our algorithm actually is evaluated by a bunch of experiments. So particularly interesting is for this one, we found this selective estrogen modulators could be inhibit this virus of Pseudomonas. Actually, but it will not kill the bacteria, just inhibit this virus. Now this are also already validating this C. elegans models. Now we proceed to test it in mouse models. So because we have this kind of genome scale target profiles, right, we predict, so we can screening chemicals based on this genome scale target profile, not just a single target. So we believe they will give you more correlations with phenotype. So here's example. Based on this phenotypic profile similarity, we can link these drugs together. All these drugs actually, based on name, you'll see this anti-cancer therapies. Bottom is kinase inhibitors, right? So this set of drugs is actually developed for totally different this disease. For example, anti-worm. So we found this high similarity between these drugs with anti-cancer drugs. So it's we carry out the cell line is assay to validate these findings. Also, there's some new chemicals. It's not in clinical trial, but we can screen them against these whole profiles. So this have promising drug leads for anti-cancer targets. After we have identified these off targets and all these targets, we can map into this biological network and link them to this, for example, in this case, this differential express genes. Then we can identify some critical nodes will correctly affect this information transmission of these drug actions. So you can think this will delete this node will have a big impact on how this drug actions, right? So we believe this kind of genetic factors could be served as these biomarkers. So again, back to structure, you can see how mutations affect drug action. And also for this kind of node, in this case, is how this phosphorylation state will change these bindings, this kind of network binding, protein-protein interaction, reveal the network. Okay, so that's a summary. So we think this kind of multi-scale modeling will give us this more mechanism understanding on the drug actions, and then provide us the critical information on this, establish this causal association between genotype and phenotype, not just like the statistical correlations. So in order to successful for this kind of multi-scale modeling, we need to integrate structural biology and system biology. It will facilitate multi-scale modeling and also give us new opportunities for drug repurposing, drug rescue, and precision medicine. So here is this work is based on these collaborations with this